Okay, so I'm making this video. This is different. This is going to just be kind of like a little study journal or, captain's log type deal for one of my classes. I'm taking a microbiology class right now, so I wanted to make this series kind of like, again, like a captain's log of, hey, you need to study this stuff. You goofball, study it later, I'll tell my future self, so just so I get a little bit more familiar with the material, I'm going to start this like journal thing, this captain's log of things you need to study so you don't fail this class. Alright, so this topic will be the brief history of microbiology. So I'm just going to write microbiohistory, kind of goofed up there. I was about to write microbiology, but then I stopped midway, so we're just going to do that. So, again, first chapter here is kind of just a review. The first week of classes, it's always just a review. But by week 2, we go out swinging, They're already doing all these crazy labs and whatnot. So let's start reviewing now while we're ahead of the curve. Okay, so it's a very relatively new field. Microbiology actually started in the 1600s, specifically that year. We have someone named Antony van Leeuwenhoek. Sorry about that pronunciation, Antony. He was making this simple magnifying glass, so Leeuwenhoek. Sorry again for the pronunciation on that last name there. And he made a magnifying glass, so a very simple one. That's just an artist's rendition, I guess, of what it would have looked like. And I think it was like a tube and it had like a holder. That's not important. He just made a simple magnifying glass. And he saw something for the first time called animalcules. So molecular animals, that's what he called these things. They're very tiny. We're talking about the first viewing of microbes in history by a scientist. So he makes all these drawings of his observations. He says there's like a rod-shaped one like that, I guess, and it moves. There's some dot ones. A lot of different samples here. He's basically looking at an assorted mixture of microbes in this first viewing of these organisms under the magnifying glass. At the same time, around this period, we also have Robert Hooke. I have a friend named Robert. He was in my classes in the last few semesters. Hope he's doing well. Anywho, this Robert Hooke, he is also credited with the same discovery in the start of microbiology. He looked at the microscopic mushroom, aka a mold, so micro-shroom. Mushroom, let me write, there we go. Okay, so this was in 1665, and he saw these, like, tiny thread-like things on a piece of bread, so he saw mold under the microscope. Alright, so why are these little dudes here and here so important? Why are they important? Microorganisms are regarded to be the foundation of all life on Earth and our whole history of life existing. Those roughly 4 billion years in the life of the world and the universe, they have been there. All of these creatures that we know today, so like plants, animals, I'm just gonna draw a bad stick horse to show animals. All of them, they came from microbes. We have the basically the same biochemistry as well as the same types of processes within our systems as microbes are, so they're very useful for studying. Alright, so how did we get microbes? Why are they here? So some of the first theories include spontaneous generation. Just like spontaneous combustion is instantly bursting into flame, spontaneous generation means the instant creation of life from non-living material. It's also known as abiogenesis, but right now we're gonna just focus on its name as spontaneous generation. People like Aristotle, that wise dude from Greek times, the Greek philosopher, he believed in spontaneous generation, where non-living things will give rise to living things. And throughout history, on our track with all of the 1600s and 1700s, and then even going into the 1800s, we had people who are supporting spontaneous generation as well as refuting it using their scientific experiences. Alright, so the first experiment that is of note comes from a guy named Francesco Redi. And he did this experiment in 1668. He was a biologist, and he wanted to check if worms that would form on rotting meat, were they there from eggs, or were they just created out of nowhere, kind of like a magic trick, you know, they just appeared suddenly. So these worms were originally thought to just appear. So, in order to test this, so he had these jars. One of them had a lid, one of them did not. We have meat at the bottom. I know that just looks like an oval, maybe possibly a fruit, but let's make it kind of like a piece of steak by doing those grill marks. There we go. It's not a dry piece of steak because it's probably not from Longhorn, but it's probably a well-seasoned steak, so something from like a Michelin star restaurant or something your parents or yourself probably made. Who knows? Joshua Wiseman may have cooked this steak. Alrighty, so we have one of the jars covered. One of them is open to the air. We have flies that are going to just land onto the meat. This is it being exposed to the outside environment. And after a while, he noticed the jar that was covered still had that beautiful Joshua Wiseman steak on there. It was still really pristine. There aren't any flies or maggots or anything like that. However, the one that was open to the environment had a ton of these maggots just flying off of it and squirming. There's squirming dudes on there. Okay, so after this experiment, he concluded, based off of the evidence, that flies do not spontaneously regenerate. So flies don't just appear. So now, the next question is, what about the microbes? Do they still appear? They're much smaller than a fly, so they're, at the time, people thought it must have been a different case for them. So, after the experiment that Redi did, we have some people trying it with microbes. In 1749, we have a guy named Needham. This guy, he really liked experimenting with different broths. So a broth is basically a liquid mixture that has all these ingredients. They're kind of like nutritional growing liquids for different scientific experiments. So he would have this broth, probably has a ton of like proteins or other substances in there important for living things, and he would boil them. So here's his fire source. We're going to just put a little flame on there, and so now it's heating up the broth. Probably had a stand like that. Okay, so he's heating this broth, right? Usually, you would do this over a Bunsen burner like we have in this image. So while he's heating the broth, he then checks it. After it cools it down, you know, he picks it up safely. And there are still microorganisms that he sees in the solution. So, after this evidence, he demonstrated that the boiling of the broth still produced microorganisms. I'm going to just put microbes. So he supported spontaneous generation with this evidence. He gave it a little check mark like we just did. The next person who did an experiment regarding to microbes as well as spontaneous generation would be in 1776, the year that the American country won its independence, actually. A guy named Father Spallanzani. He contradicted Needham's results. So he looked at Needham's experiment here, and his conclusion, he said, hey, something is sus about this experiment. There must be something else going on. Sorry about that. That was such a rude notification there. So he looked at that, and he thought that was sus, like extremely sus. So what he did, he set up the same experiment. He had his broth, again on an open flame or a heat source, and this time he burned or boiled the broths longer. So and then after he boiled them, he sealed the flask off using some glass or melting the neck of the glass. So kind of like that. So that's a melted, sealed container now with his broth inside. After this, he discovered that the broths remained sterile. So there wasn't any contamination or anything like that. There was nothing living in the broths after he stored them away and sealed them off. So he kind of threw shade at Needham's results here, saying that you just had to boil them longer and then seal it off, no microbes. So this experiment kind of disproved spontaneous generation. However, there's more of a controversy as we later on. There's more people who still found microbes after boiling. They thought that there's something called the vital force that they're trying to mess around with. And some of the people still believing in spontaneous generation, they believe this vital force may have survived the temperatures or something like that to lead to microbes. However, this will be disproven by an experiment 100 years later in 1876 when a guy named Ferdinand, not the bull from that movie, that animated movie, but Ferdinand Kahn, he would go on to discover something called an endospore, which is something that bacteria are able to develop in unfavorable conditions. So when there's extreme temperature or low nutrients, some bacteria will make this very difficult and rigid fortress around themselves called an endospore. They would make these endospore, and inside they would be dormant. So here's an endospore. As you can see, it's dormant, it's heat resistant. I guess there's like a heat source there blasting it with a flamethrower as if it was a Charizard melting something, but it's heat resistant. The fire doesn't kill the endospore. So this may have been why boiling the broths in Needham's experiment all the way, if we go back over here, maybe that's why these microbes are still there after the solution was being boiled, because of the endospore. Alright, now we're in kind of the VIP section of the whole modern microbiology sphere. We go over here and we meet Louis Pasteur. He was a French chemist, and he's known as the father of modern microbiology. This is where we get pasteurization, where we have all these products such as milk. Here's a very badly drawn carton of milk. And you'll see on the label it says pasteurized. And this procedure is actually named after Louis Pasteur, and we'll see why in a bit. So a lot of his experiments, they demonstrated how the outside, the external air, as well as just surfaces, are filled with microorganisms. So here's the air, I guess, like, you know, from the Avatar anime. So there's like the air symbol. I guess I'll put it in there too. I don't know what it actually looks like. So let's say Aang is here and he's blowing all the air, you know, as oh shoot, sorry. Has an air on his forehead like that. So he's blowing all this air, right? So Louis Pasteur, he takes the air that's just in the surroundings. He filters the air through a cotton plug, kind of like this. So there's the plug, and then here's the fluffy cotton ball. So that air is going through this filter. And he trapped the microorganisms. You saw there a ton from just the air. Another one of his experiments directly looked at spontaneous generation. So he had something called a swan neck flask. Where it's basically a regular flask, you know, you have a bulb, you have some broth at the bottom, it's over a heat source. However, like a swan, it has this cool neck thing going on like that. Right, so what Pasteur did with this flask is that he boiled the broth at the bottom, and the air escaped out as he was doing so. Over time, there's some microorganisms that just started settling right here, in this section of the flask. Those are microbes. Then, after a while, a lot of time passed, so I'm just going to say time there, kind of like, you know, those Pokemon evolution charts. So over time, we took the flask with all the broth. So Louis Pasteur took the flask, tipped it on its side. So now, that region where the sterile broth was boiled and stuff, now it's dipping into the area that had those microbes. So we're going to, again, label where all those microbes were festering. They're having a little house party. The broth was tipped so it could touch all those microbes and sweep them back and forth. You know, they get mixed in the broth like so. And then, after a few hours, as well as several days, the broth that was once sterile now had all these little buddies inside of it, had all these tiny microbes in it. So, what did Pasteur's experiment appear to show? Well, boiling the infusions remains sterile, even though they're open to the air, because you have this separation. Boiling broth remained sterile, even though it had this opening. This is open to the air. As you could see, the air could escape from boiling the broth, as well as air could enter. Any of those microbes that we saw earlier from this experiment with the filter, they would have ended up in the neck of the flask here, in this pink region, so it would remain sterile. Though it was open. And the only way he got the broth to become contaminated was when the flask was tipped over, and again, those little buddies were able to get into the liquid when it was tipped over from their spot in the neck. So that's how you led to the multiplication of all the bacteria in the broth that was once stable. So since the tipping of the flask introduced microbes into the broth, this disproved the vital force was necessary for bacteria to enter. So basically, Pasteur, he was like Dorothy and, the Lion, the Knight and stuff. They went to the Wizard of Oz of spontaneous generation. They looked past the curtain. No, the bacteria have to come from bacteria, or a source of bacteria that was already there in order to appear in the broth. So the Wizard of Oz, the curtain was pulled back. There was no vital force at all. Okay, after Louis Pasteur's experiments, we get into something called the germ theory of disease. And this took place during the golden age of microbiology. In this part of the theory, the microbes were actually the cause of many diseases. Instead of bad air or, oh, that's just how things are, they just appeared. No, now we're saying after all these experiments, microbes cause diseases. And by the end of the 1800s, so in the 1900s, by that time, most pathogenic bacteria were ID'd. We knew who they were, so we ID'd a ton of pathogen bacteria. We also started looking at virology, or the study of viruses. And we understood that we needed control of efforts to stop some of these pathogenic bacteria. So, control efforts that were started in the 1900s. So we had antibiotics. These kill, of course, the infectious diseases and the bacteria that cause them. So antibiotics are for bacteria and the diseases that their infections cause. Then we also have vaccines to prevent viruses or the disease that viruses create and cause. Also in microbiology, we get into different fields such as the ecology, ecological principles of it, as well as applied in other industries, such as medicine, food, such as beers made by yeast. Ecological benefits include cycling of nutrients. We wouldn't survive without microbes. They're constantly breaking down materials, constantly breaking down waste, turning it into useful molecules for other applications in life, as well as in the industries, such as the production, again, of food, medicine, vitamins, as well as useful chemicals that we could use as pesticide or degradation of waste again. We also have biotech. Sounds extremely fancy. This is where you're going to apply techniques seen in biology to solve problems. We also have genetic engineering, which is possible thanks to microbiology. So introducing genes into organisms, disease-resistant crops. So like the banana, we're in dreadful need of another banana to be found or developed, because all the ones right now, the type that are right now, they're all clones of each other. Once there's like a pathogen that gets rid of all those bananas, so this, like, let's say this is the species of banana they have right now, right? Let's say there's a pathogen that's just really giving them a bad day. As you can see, there's the pathogen going over there and blasting that banana to the shadow realm. We need to probably develop a new disease-resistant banana crop, let's call it B, before all the clones, all the bananas, are also affected by that pathogen. That's why it's important to sometimes create these disease-resistant crops, such as this example with the banana. Also, medicine could be produced by bacteria using genetic engineering. Insulin is actually harvested from E. coli, which are programmed through genetic means to create the proteins used for insulin. So your insulin is actually coming from E. coli. Genome sequencing, this is another big one. This is like Iron Man level craziness that we're going into right now with all the biotech and microbio stuff. Some pretty cool, almost like fantastical stuff are happening day by day in this field. So this allows for the detection of different microbes and possibly new ones too. And we could also look at important genes for study. Right, challenges. Diseases are, of course, still a present challenge today. We've went through a pandemic. COVID is still around, just different variants of it. A lot of pandemics. There's also superbugs. These are bacteria that are being selected for the genes that are giving them resistance to certain antibiotics by the overuse of antibiotics. So let's say bacteria A. This is bacteria A. Everyone say hi to bacteria A. Let's say that we use this chemical. Let's say it's Professor Utonium's chemical X, right? So we're going to use that on the bacteria. We have 1, 2, 3, 4, 5, 6, 7 of them. So let's say we keep using chemical X. So let's say chemical X first kills 4 of the bacteria, right? Leaving only 3 who are just by chance resistant to it. They're resistant to it because they so happen to have a gene that codes for a protein that gives them resistance to chemical X. Okay, so if we just keep using chemical X on the bacteria without changing the antibiotic, it's not going to do anything. Instead, this bacteria here, bacteria A, they're just going to multiply. There's nothing stopping them from multiplying. This antibiotic, they're just going to, again, be resistant to it because they were previously resistant to it by chance because they had that gene that gave them a resistance when it was expressed. If we, however, treated bacteria A after this initial dosage, if we treated them with bacteria killer Y, let's say this is chemical Y, right? They're not resistant to chemical Y, so instead of using chemical X, we switch to this other antibiotic, or we switch it to a different dosage. We just need to change the chemical that we're going to be using. We're going to change or use a different antibiotic. We get 0 bacteria. However, unfortunately, in many cases, this is the path that we're going. We are running out of antibiotics to choose from, And we are unfortunately selecting for resistance over and over and over again, letting these resistant bacteria proliferate because we aren't using new antibiotics. We're overusing the ones that we know. So that's why there's a superbug crisis right now in both the medical as well as in the biological aspect of microbiology. Alrighty, so that was like a run through of all that microbio basics, history of microbiology. Again, I want to tell my future self, please study this. I made a fun video for you. Hopefully the content is good for the final and whatnot. So yeah, thank you so much for tuning in. Please do something nice for someone.